Welcome to the last chapter. It is nutrition in plants and animals. Now, all living organisms require food to sustain life. Food is also required for carrying out metabolic activities such as respiration, growth, repair, and maintenance. The process by which organisms obtain nutrients and assimilate them is referred to as nutrition. Nutrition is the process by which organisms obtain and assimilate nutrients. The modes of nutrition differ depending on the type of organism. Both autotrophism and heterotrophism are modes of nutrition that have been identified among living organisms. Autotrophism is a mode of nutrition in which the living organism manufactures its own complex food substances from simple substances. These simple substances are carbon dioxide, water, light, and chemical energy. The simple substances are obtained from the immediate environment. Living organisms that make their own food are known as autotrophs. Heterotrophism is a mode of nutrition that involves taking complex food materials such as carbohydrates, proteins, and fats obtained from the bodies of plants and animals. Organisms that feed on already synthesized foods are called heterotrophs. Autotrophism. Autotrophism is a process by which some plants manufacture food substances such as carbohydrates from simple substances such as carbon dioxide and water. Where sunlight is used as the source of energy, the process is referred to as photosynthesis. Some non-green plants obtain energy for making food from oxidation of chemicals. The process of manufacturing food is therefore known as chemosynthesis. As a result of photosynthesis, food substances such as carbohydrates, proteins, and fats are formed. Plants are therefore the primary producers, and animals directly or indirectly depend on them as a source of food. Therefore, it can be concluded that the process of photosynthesis involves the conversion of light energy into a form that can be utilized by all living organisms. During the process of photosynthesis, carbon dioxide from the atmospheric air is used. Oxygen is given out and released into the atmospheric air as the byproduct. Animals and plants use oxygen from the atmosphere for respiration. They release carbon dioxide as one of the products of respiration. This way, photosynthesis helps to maintain the level of these two gases fairly constant in the atmosphere. The process of photosynthesis occurs in green plants, mainly in the leaves. In order to understand the process of photosynthesis, it is necessary to study the external and the internal structures of the leaf. External structures of a leaf. A leaf is a thin, flattened organ which is attached to the stem or a branch of a green plant. Typical simple leaf consists of a flattened surface called the lamina. The lamina is green in color and contains the photosynthetic tissue. In dicotyledonous plants, simple leaves have thick midrib which runs in the middle. From the midrib arises small veins that run into the lamina, forming an extensive network of veins. In monocotyledonous plants, the midrib is absent but small veins run parallel to each other. Internal structures of a leaf. A transverse section of the leaf examined under a light microscope reveals the internal structures appearing on your screen. The following is a description of the structure and functions of the parts. Cuticle. This is a thin, non-cellular, waxy, waterproof, and transparent layer that coats the upper and lower surfaces of the leaf. It reduces excessive loss of water and protects the inner tissues of the leaf from mechanical damage. It also protects entry of disease-causing microorganisms. The cuticle is transparent. It allows penetration of light for photosynthesis. Epidermis. Epidermis is a thin tissue, usually 1 cell thick, on the upper and lower surfaces of the leaf. The epidermal cells have no chloroplast except the guard cells. Chloroplasts are the organelles that contain photosynthetic material known as chlorophyll. The epidermis secretes the cuticle and also protects the inner tissues from mechanical damage and entry of disease-causing microorganisms. Palisade layer. Palisade layer is a layer of cells located beneath the upper epidermis. It consists of cylindrical shaped cells closely packed together and with a long axis perpendicular to the surface. They have numerous chloroplasts containing chlorophyll, which is necessary for photosynthesis. Their position and arrangement allows them to receive maximum sunlight. The spongy mesophyll layer. This is a layer of cells between the palisade and the lower epidermis. The cells in the spongy mesophyll layer are irregularly shaped and loosely arranged, creating large air spaces in between them. The air spaces provide communication pathways through which gases diffuse in between the cells. Unlike the palisade cells, these cells contain fewer chloroplasts. This explains why the lower surface of the leaf is lighter in color than the upper surface. The guard cells are special bean-shaped epidermal cells mostly found on the lower surface of the leaf. Unlike the adjacent epidermal cells, guard cells contain chloroplasts which are able to carry out the process of photosynthesis. They control the opening and closing of the stomata. They also control water loss. Through the stomata, air moves in and out of the leaf. Leaf veins. The leaf has an extensive system of veins which ensures that every cell is close to the supply of materials. Each vein is a vascular bundle which consists of xylem and phloem tissues. Through the xylem, water and mineral salts are conducted from the roots to the leaf cells, while manufactured food is translocated through the phloem from the leaf cells to the rest of the plant. Now, leaves possess special structural features which enable them to carry out the process of photosynthesis efficiently. Adaptations of the leaf to its photosynthetic function. The structural features include the following. The leaf has a broad, flat lamina which provides a large surface area for the absorption of carbon dioxide and sunlight. The thinness of the leaf allows light and carbon dioxide to pass through a short distance to reach the photosynthetic cells. The presence of stomata ensures efficient diffusion of carbon dioxide into the leaf. The cuticle and epidermis are transparent enough to allow penetration of light to the palisade cells. The palisade cells themselves contain large number of chloroplasts, and the arrangement and location next to the upper epidermis enables them to receive maximum sunlight. The existence of extensive veins which conduct water and mineral salts to the photosynthetic cells and removes the products of photosynthesis. The air spaces in the spongy mesophyll layer are large enough to allow gases to circulate easily. Finally, the regular arrangement of leaves on the stem minimizes overlapping and overshadowing, and this is called leaf mosaic. Chloroplasts. Chloroplasts are disc-shaped organelles found in the cytoplasm of some plant cells. Each chloroplast is bound by a double-unit membrane, the outer and inner membranes. Internally, the chloroplast consists of layers of membranes known as lamellae suspended in an aqueous matrix called stroma. The lamella may, at certain intervals, form several layers of membranes grouped together to make a granum. Chlorophyll molecules and other photosynthetic pigments are located in the granum, which provides a large surface area to accommodate a large number of chlorophyll molecules. Chlorophyll molecules absorb light energy, which is necessary for photosynthesis to take place. The stroma contains enzymes that speed up the process of photosynthesis. The process of photosynthesis. Now, the raw materials for photosynthesis are carbon dioxide and water. For the process to take place, it requires the presence of light and chlorophyll pigment. As mentioned earlier, the process of photosynthesis occurs in the chloroplast. Water and carbon dioxide molecules undergo several chemical processes in the presence of sunlight to form carbohydrates, and oxygen is given out as a byproduct. The process is, however, more complex than what is represented in the simple series of equations given. It involves chemical reactions that can be outlined in two general stages, the light and the dark stages. The light stage, also known as light-dependent stage. Now, this is the initial stage in the process of photosynthesis, and it occurs in the grana of the chloroplast. The chlorophyll molecule absorbs light energy, which is used to split water molecules into oxygen and hydrogen atoms, and this process is called photolysis of water, and it's shown by the equation on your screen. The hydrogen atoms that are produced by the splitting of water enter the dark stage, while oxygen is released to the atmosphere or used by the plant for respiration. Some of the solar energy absorbed by chlorophyll molecules is used in the formation of energy-rich adenosine triphosphate, ATP, which is later used in the dark stage. This reaction involves conversion of light energy into chemical energy. The dark stage, also called light-independent stage. This is the second stage of the process of photosynthesis. It proceeds whether light is present or not. This process involves combination of carbon dioxide with hydrogen atoms to form simple sugar, such as glucose. This process is known as carbon dioxide fixation, which involves a series of enzyme-controlled reactions as shown on your screen. The equation on your screen is a summary of carbon dioxide fixation. The energy required for this reaction is produced by the ATP from the light stage reaction. The immediate product undergoes further reactions to form simple sugars such as glucose. Some of the glucose formed is directly utilized by the plant cells, while the rest is converted to starch and stored in the plant. In addition to glucose, fatty acids and amino acids are also formed in the dark stage of photosynthesis. The main factors which affect the rate of photosynthesis are light intensity, carbon dioxide concentration, temperature, and water. Light intensity. Now, the intensity of light varies from day to day and from place to place. On a bright sunny day, plants photosynthesize faster than on a dull cloudy day. The rate of photosynthesis increases linearly as the light intensity increases up to a certain level, when it slows down and finally levels off. Extremely bright sunshine may actually damage plants because of high amount of UV light. It is important to note that the quality of light also affects the rate of photosynthesis. Most plants require red and blue wavelengths of light for photosynthesis. Plants growing in shaded areas receive low quantities and poor qualities of light, which slows the process of photosynthesis. Carbon dioxide concentration. Now, the amount of carbon dioxide in the atmosphere is about 0.03% and does not vary very much. Under controlled conditions like greenhouse, an increase in carbon dioxide concentration results in a linear increase in the rate of photosynthesis up to a certain level when it slows down and finally levels off. Temperature. Photosynthesis is a process controlled by enzymes. Now, enzyme-controlled reactions are affected by temperature changes. At low temperature, the rate of photosynthesis is low. A rise in temperature by 10 degrees results in the doubling of the rate of photosynthesis up to 40 degrees Celsius. Further increase of temperature decreases the rate sharply and finally stops. This is because the excess heat destroys enzymes which are responsible for the chemical reactions. Now, for each factor considered, the rate of photosynthesis increases up to a point where it levels off. At this point, there is a limiting factor. For example, where the graph levels off for carbon dioxide, the limiting factor may be light intensity. If this were increased, the rate of photosynthesis would increase beyond the plateau. Water. Plants need water for photosynthesis. A plant whose leaves are withering due to lack of water may photosynthesize at a lower rate. However, water is needed for many other purposes besides photosynthesis, and the effect of water shortage on photosynthesis may be indirect. To maximize the rate of photosynthesis, optimum temperature conditions with adequate rainfall and sufficient carbon dioxide are necessary. Next is chemicals of life, and these are chemical compounds which constitute living organisms. Now, cells, tissues, and organs are composed of chemicals which we call chemicals of life and are obtained from the environment. The study of chemical compounds found in living organisms and the reactions in which they take part is known as biochemistry. The study attempts to review the chemical composition of living systems. Many of these chemicals are identical to those found in non-living organic matter. However, we shall consider carbohydrates, lipids, and proteins. Carbohydrates. Carbohydrates are compounds of carbon, hydrogen, and oxygen. The elements are in the ratio of 1 to 2 to 1. This gives the carbohydrates a general formula of CH2O. In bracket N, where N represents the number of carbon atoms a molecule of a carbohydrate has. Carbohydrates can be divided into the following types: monosaccharides, disaccharides, and polysaccharides. Monosaccharides. Monosaccharides are the simplest carbohydrates and have the general chemical formula of CH2O into bracket N, where N is 6, and hence their chemical formula is C6H12O6. Examples include glucose, fructose, and galactose. They are called simple carbohydrates. Properties of monosaccharides. Monosaccharides are soluble in water to form a sweet-tasting solution. Sweet-tasting fruits owe this property to the abundant content of monosaccharides in them. When monosaccharides are mixed with Benedict solution and heated, the copper sulfate is reduced to red copper 1 oxide. Thus, They are described as reducing sugars. They are crystallizable. Molecules of monosaccharides link together to form complex carbohydrate molecules, and this process is called condensation. In this reaction, water molecule is also formed. For example, two glucose molecules can be condensed and linked together to form one molecule of disaccharide. Functions of monosaccharides. Now, monosaccharides are oxidized during respiration to release energy. Glucose is the most common respiratory substance and the most common monosaccharide. When condensed together, monosaccharides form polysaccharides such as starch and cellulose in plants. Glucose can be converted into glycogen, which is stored in animal tissues.
Okay, food tests for reducing sugars. You need a test tube rack, 2 test tubes, one for the control, one for the actual experiment. You need some glucose, 10 ml solution, glucose solution. You need 10 ml of water, and what we call the Benedict solution. So, you add 10 ml of water, you need 10 ml of glucose solution. You add a few drops of Benedict solution. You stir it a bit, and if you want to see the actual experiment, you need to put it in a water bath at temperature 70 degrees. So this is a positive test. Here you are in presence of glucose, and here's your control.
Disaccharides. Disaccharides are formed by linking two monosaccharide molecules through the process of condensation. Therefore, they are more complex than monosaccharides. Their formation from two monosaccharides results in the loss of water. For example, glucose condenses with another glucose molecule to form maltose and water. Glucose plus fructose forms sucrose and water. Galactose condense to form lactose and water. Now, from these equations, it can be concluded that the type of disaccharide formed depends on the monosaccharide unit that enter into its composition. Properties of disaccharides. Like the monosaccharides, disaccharides are soluble in water, forming a sweet-tasting solution. Sugarcane juice is rich in sucrose. This juice is processed to make ordinary sugar. Some disaccharides such as sucrose cannot reduce copper 2 sulfate in the Benedict solution unless they are first broken down into their constituent monosaccharides. They are therefore known as non-reducing sugars. However, some disaccharides such as maltose reduce copper 2 sulfate in Benedict solution when heated together. They are known as complex reducing sugars. Disaccharides can be readily broken down into their constituent monosaccharide molecules in a process known as hydrolysis. Hydrolysis is the reverse of condensation. This process involves splitting the complex molecules in the presence of water. In nature, disaccharides are hydrolyzed by enzymes. In the laboratory, hydrolysis can be carried out by heating them with an acid such as dilute hydrochloric acid. Functions of disaccharides. Now, disaccharides are hydrolyzed by enzymes into monosaccharides, which are then oxidized to release energy.
Okay, here's the food test for non-reducing sugar. You need test tube rack, 3 test tubes, you need some cylinders, you need some, again, Benedict solution, hydrochloric acid, sodium hydroxide, and some sucrose solution. Okay, let's add 10 ml for our test control, the water. Then we have our sucrose solution, 10 ml. And we're going to add a few drops of hydrochloric acid. Test control. This one we don't put any hydrochloric acid, we'll leave it aside. We're going to take those 2 test tubes and we put it in the water bath for a few minutes. You put your 2 test tubes, test control, and the sucrose or sugar solution in the water bath for a few minutes. While they're cooling down, you have to neutralize the hydrochloric acid, so you need to add a few drops of sodium hydroxide, so it should be 10 drops. Again, you don't touch that one here. Now it's time to add the Benedict solution. Add a few drops in each test tube. And we take those test tubes and we put them in the water bath for a few minutes. Okay, so we did the Benedict test with non-reducing sugar. As you can see, this is a control with water, so no reaction. Here, this is the test tube with sugar and hydrochloric acid, so at the end you get a reaction because you broke down the disaccharide into glucose and fructose. And that one is basically sugar, so sucrose, with no hydrochloric acid.
Polysaccharides. Polysaccharides are made up of many molecules of monosaccharides. Thus, their molecules are long and more complex. They have a general formula of C6H10O5 into bracket N, where the value of N is very large. The polysaccharides commonly found are starch and cellulose in plants, while glycogen is found in animal tissues. Starch. Starch is present as stored food in plants. It is formed by condensation of large number of monosaccharides. Plants that are rich in starch include maize, wheat, potatoes, and rice. These plants are grown as sources of food. Cellulose. Cellulose exists as a compound of the cell wall in plants. It is made up of more molecules of monosaccharides than in starch. Cellulose gives the plant cell their definite shapes. Glycogen. Glycogen is present as stored carbohydrate in animal tissue. Glycogen is synthesized from excess glucose. Polysaccharides are known as non-sugars. All of them are insoluble in water and do not have a sweet taste. Now, the next category of the chemicals of life are the lipids. Now, these are the fats and oils. Fats are found in animals, whereas oils are found in plant leaves, fruits, and seeds. However, a few animals such as whales contain oil. Like carbohydrates, they are compounds of carbon, hydrogen, and oxygen. However, the number of oxygen atoms are fewer in lipids than in carbohydrates. The building units of lipids are the fatty acids and glycerol. The synthesis of lipids molecules require 3 fatty acid molecules condensing with 1 molecule of glycerol. Now, fatty acids are of different types. Therefore, the nature of a lipid depends on the fatty acid it contains, even though the glycerol is the same in all lipids. Like carbohydrates, complex lipids are also formed by condensation process. Examples of complex lipids include phospholipids, waxes, steroids, and cholesterol. Properties of lipids. When fats are heated, they readily change into liquids, while oils solidify if subjected to low temperatures. Both fats and oils are insoluble in water. However, they will readily dissolve in organic solvents such as alcohol, ether, and chloroform. When fats and oils dissolve in these organic solvents, they form emulsions and suspensions. Lipids are quite inert, a property which enables them to be stored in the tissues of organisms. The presence of fats and oils in a substance can easily be detected by using Sudan 3 dye, grease spot test, and emulsion test. Functions of lipids. Lipids are sources of energy just like monosaccharides. A given weight of lipid will liberate almost twice as much energy as an equivalent weight of a monosaccharide. Lipids as a source of metabolic water. And oxidized lipids release more water and energy than monosaccharides. Such water is known as metabolic water, which supplements the requirements in the body. This explains why some animals such as camel accumulate large quantities of fat reserves in their body. They metabolize these fats to obtain the metabolic water in addition to the energy released. Lipids as structural compounds. Now, lipids are constituents of plasma membrane, and oils are storage materials in some seeds such as groundnuts, castor seeds, maize grains, and so on. Lipids used for heat insulation. In animals, fat is deposited under the skin where it forms the adipose tissue, which acts as a heat insulator. In this way, fats assist to reduce the loss of heat from the bodies of mammals. Mammals living in temperate regions have thick adipose tissues, which generally reduces heat loss. In addition, the thick adipose tissue in some aquatic animals helps them to be buoyant in water. Protection. Fats are deposited around the major organs such as the kidney, heart, and at the back of the eyeball, where it acts as a shock absorber. The last category of chemicals of life are the proteins. Now, proteins, like carbohydrates and lipids, are compounds of carbon, hydrogen, and oxygen. However, unlike carbohydrates and lipids, they contain also nitrogen and sometimes phosphorus or sulfur or both. Proteins are made up of small units called amino acids. There are about 20 different types of amino acids occurring naturally. All amino acids contain amino group, which consists of hydrogen and nitrogen, but the number of carbon atoms differ from one amino acid to the other. Proteins are referred to as nitrogenous compounds because of the presence of nitrogen in their structure. A single molecule of protein can have hundreds or even thousands of amino acid units which are joined together. Like carbohydrates, proteins are also formed by condensation. The process involves combination of two amino acids to form a dipeptide molecule. The bond between the two amino acids is called a peptide bond. During this process, a water molecule is formed. Continued condensation leads to the addition of more amino acids to a protein chain. A long protein chain known as polypeptide results. The uniqueness of a particular type of protein is determined by the type and sequence of the amino acids that it contains. Theoretically, the possible number of ways in which amino acids could combine may lead to an infinite number of proteins. However, this is not usually the case since organisms possess mechanisms which control the synthesis of specific protein, and hence the number of proteins in life is much lower than is expected. Properties of proteins. Now, most proteins dissolve in water but do not form a true solution. They only form colloidal suspensions where the particles remain suspended in water. Most proteins are denatured at temperatures of 40 degrees Celsius. This changes chemical and physical properties of proteins and hence their physiological activity. Denaturing can also be caused by chemicals such as acids, bases, detergents, and organic solvents. Denaturing is irreversible, hence it's a permanent damage to the protein in the tissue. Proteins have both acidic and basic properties. Due to these properties, proteins are described as amphoteric. They therefore react with acids and bases. This property enables proteins to combine with non-protein compounds to form conjugated proteins. For example, in mucus, the non-protein compound is a carbohydrate, while in hemoglobin, the non-protein compound is iron. Functions of proteins. Structural functions. Plants synthesize amino acids which build up the proteins. Animals obtain the proteins from the plants and then break them up during digestion and use the amino acids to form animal protein. In both plants and animals, proteins are used to make the framework of living systems. For example, the plasma membranes, connective tissues, muscle fibers, hair, hooves, nails, and skeletal material. Proteins as metabolic regulators. Now, enzymes and some hormones are protein in nature. Enzymes are organic catalysts which speed up metabolic reactions and thus they control the rate at which metabolism takes place. Hormones, on the other hand, are chemical messengers which act as regulators of many body processes, for example, growth, reproduction, regulation of chemical substances such as salt, water, and sugar. Proteins are sources of energy. Like carbohydrates and lipids, proteins can be broken down to release energy. However, amino acids cannot be stored in the body like carbohydrates and fats. Proteins are normally used as sources of energy in conditions of starvation. Enzymes are biological catalysts which are protein in nature. They are produced in living cells, and as catalysts, enzymes speed up or slow down the rate of chemical reactions in the body without themselves being used up. Next is nutrition in animals, and we start with heterotrophism. Now, heterotrophism is a mode of nutrition that involves taking in complex food materials such as carbohydrates, proteins, and fats obtained from the bodies of other plants and animals. Organisms that feed on these already synthesized foods are called heterotrophs. The heterotrophs break down or digest these foods into simple soluble molecules which can pass through the cell membrane during absorption. Heterotrophs obtain their food using the following heterotrophic modes of nutrition. 1. Holozoic. This is a type of nutrition characteristic of animals where solid complex substances are ingested, digested, and assimilated in the body. Saprophytism. This is a type of nutrition where organisms obtain nutrients from dead organic matter, causing decomposition. Parasitism. This is an association where one organism, the parasite, feeds on or obtains nutrients from the tissues of another living organism, the host. Symbiosis. It is a type of association where two organisms live together and mutually benefit from each other. Heterotrophs are dependent on autotrophic organisms as a source of preformed organic materials. Many big-sized animals take in large particles of food of different types and are therefore grouped into three categories, namely herbivores, carnivores, and omnivores. These animals have four different types of teeth: the incisors, canines, premolars, and molars, and are thus referred to as heterodonts. Other vertebrates have teeth of the same sizes and shapes and are therefore called homodonts. The description of types of teeth, their arrangement and specialization is called dentition. Teeth are arranged in groups to occupy specific positions in the jaw. The incisors are flat chisel-shaped with sharp-ridged edge for cutting and biting food. They have one root. Canines are conical with sharp-pointed tips, especially modified in carnivores for seizing prey and tearing flesh. They normally have one root. Premolars have two roots, while molars have two or more. Both have broad surfaces and may be ridged with cusps on their crown for crushing and grinding of food. Dental formula describes the number, type, and position of the teeth in the jaw of mammals. The number of teeth recorded represent the half-total teeth in the upper and lower jaws. The names of types of teeth are abbreviated as follows: incisors as I, canine as C, premolars as PM, and molars as M. The dentition determines the mode of feeding, whether herbivores, carnivores, or omnivores. Herbivores are animals which feed exclusively on vegetation, and they are grouped as grazers or browsers. Grazers, for example cows, donkeys, and zebras, feed on grass, while browsers such as giraffes, goats, and antelopes feed on shrubs and herbs. Herbivores have several adaptations on their dentition that suit them for the kind of food they take in. Most herbivores do not have the upper incisors, but instead have a honeypad against which grass is pressed and cut by the lower incisors. The long tongue assists in the cutting and turning of grass and also moving the food during grinding. These functions are made possible by the diastema, which is a gap in the lower jaw separating the canines from the premolars. The jaws move side by side to enable premolars and the molars to grind the food. Carnivores are animals which feed exclusively on flesh. Most carnivores are hunters. Some are adapted to fast running and have well-developed leg muscles that facilitate their movement. The cheetah is one such animal that moves very fast. They also have strong jaws and sharp teeth to grasp their prey. They have four different types of teeth. The incisors are chisel-shaped and closely fitting to seize the prey. They are used for gripping and stripping flesh from the bone. Canines are long, conical, and curved to hold, kill, and tear the prey. Premolars and the molars are small except for special premolars in the lower jaw and the upper jaw, which are modified into carnassial teeth. The carnassial teeth have smooth sides and sharp edges to slice through flesh and crush bones. Jaws are attached to powerful muscles that move the jaw up and down by snapping. Structure of a tooth. Now, externally a tooth is made up of three regions. These are the crown that projects above the gum, The root, which is the part that is embedded in the jaw, and the neck, the region between the crown and the root. The crown is covered with a hard non-living layer called enamel that is made up of calcium phosphate and carbonate. The enamel is a protective covering of the tooth that also forms a surface for biting and grinding of food. Internally, a root is mainly composed of the dentine, which is not as hard as the enamel. The dentine extends to form the root and consists of living cells. Within the dentine is a pulp cavity containing nerves and blood vessels. The blood vessels provide nutrients to the living tissues in the dentine and remove waste product. Nerves detect heat, cold, and pain. The root is fixed into the jawbone by cement that is surrounded by supporting fibers. Digestive system in animals. Now, the digestive system is made up of the alimentary canal and it's associated with organs such as the liver and pancreas. The main functions of the digestive system are digestion and absorption, apart from others like ingestion and egestion. The alimentary canal, also known as the digestive tract, is a tube leading from the mouth to the anus. It consists of the esophagus, stomach, small intestine, which comprises duodenum and the ileum, and the large intestine, the cecum, appendix, colon, and rectum. The alimentary canal is relatively long and greatly folded to increase the surface area for digestion and absorption of food. It is also wide and narrow in some parts, and in others it is sac-like. Although each different portion of the mammalian digestive tract has its own adaptation in relation to function, all conform to a basic common structure. Also of interest to note is that although the digestive tract of mammals show basic fundamental plan, there exist slight variations which correlate to the kind of diet each animal eats. We now look at the food processing along the digestive tract, and this is the treatment which the food undergoes as it passes along the tract so that food nutrients are availed to the body tissues. Since much of the food an animal takes in or ingests is made up of large complex molecules, for example carbohydrates, proteins, and fats, it is first broken down to simpler and soluble molecules before they can be absorbed. The process of food breakdown is called digestion. However, small molecules like those of vitamins, mineral salts, and water are directly absorbed into the bloodstream without undergoing digestion. Along the digestive tract, food digestion occurs in the following regions, that is the mouth, the stomach, duodenum, and ileum. Each region is adapted to its function in a variety of ways. Digestion in the mouth. The introduction of food into the mouth is called ingestion. In the mouth, food is mechanically broken down by the teeth in a process called chewing and grinding. This is also known as mastication. Mastication reduces food to small particles and also increases the surface area for enzyme action. During this process, the food is moistened and mixed with saliva secreted from the salivary glands. The glands are located in various places around the mouth region, and there are 3 of them. The sublingual salivary glands are beneath the tongue. The submandibular salivary glands are under the jaw. And the parotid salivary glands are found in the cheeks in front of the ears. All these glands have ducts that open into the mouth and through which saliva flows into the mouth. Saliva is a mixture of water, mucus, and an enzyme called salivary amylase or ptyalin. The mixture is slightly alkaline, and this provides a suitable environment for the function of the salivary amylase. Water acts as a solvent while mucus lubricates the food and the lining of the mouth. The enzyme digests starch in the food to maltose. The tongue rolls the food into round masses called boluses. The tongue then pushes the boluses to the pharynx at the back of the mouth. This is the beginning of swallowing. The soft palate is raised to open the gullet and close the nasal cavity, while the epiglottis relaxes to close the windpipe or trachea. The food then passes down into the esophagus or gullet. The gullet wall contains circular and longitudinal muscles. The passage of the bolus down the gullet is aided by a wave of muscle contractions called peristalsis. This produces a series of constrictions that squeeze the bolus down the gullet and into the stomach. Digestion in the stomach. The bolus enters the stomach through a muscular valve known as the cardiac sphincter. The stomach wall is made up of thick circular muscle and longitudinal muscle layers. These muscles contract and relax, producing movements that mix the content of the stomach. The mixing is known as churning and results in the formation of a fluid called chyme. The presence of food in the stomach stimulates the secretion of the hormone gastrin, which in turn stimulates the production of gastric juice from the gastric glands embedded on the glandular layer of the stomach wall. The gastric juice contains the following enzymes: pepsinogen, which is activated to pepsin enzyme in the lumen of the stomach. Pepsin breaks down protein into peptides. Renin enzyme, which digests the protein casinogen in milk to casein or curd. Renin is abundant in suckling young children. Hydrochloric acid provides an acidic medium for the optimum activities of these enzymes. The acid also kills some bacteria that may be present in the food. Mucus, which forms a protective barrier to the stomach wall against corrosion by hydrochloric acid. The food then passes from the stomach to the duodenum, the first section of the small intestine. Digestion in the duodenum. The duodenum is the first part of the small intestine where most of the digestion takes place. Duodenum receives secretions from the following organs. The liver has specific cells which secrete the bile into the gallbladder to be stored. The gallbladder releases the bile into the duodenum through the bile duct. The pancreas lies just below the stomach. It is a thin, flat, cream-colored gland. It plays two major roles: secretion of hormones and secretion of digestive juices. The arrival of food into the duodenum stimulates secretion of the hormone secretin from the pancreas and cholecystokinin from the duodenal wall. Secretin stimulates the secretion of pancreatic juice into the duodenum, whereas cholecystokinin stimulates the secretion of bile from the gallbladder. The pancreatic juice is alkaline and contains 3 digestive enzymes. Pancreatic amylase, which speeds up the breakdown of the remaining starch into maltose. Trypsin, which digests protein into peptides. This enzyme is secreted in its inactive form, trypsinogen. Pancreatic lipase digests lipids to fatty acids and glycerols. Also produced by the pancreas is the sodium hydrogen carbonate, which neutralizes the acidic chyme and creates a suitable alkaline medium for pancreatic and intestinal enzymes. The bile contains bile salts which include sodium glycocholate and sodium taurocholate. These two salts aid in the breakdown of fats into tiny fat droplets to increase their surface area for digestion. This breakdown is known as emulsification. The salts also provide an alkaline medium in which the enzymes listed above work best. They also neutralize acidic chyme from the stomach, which contained hydrochloric acid. Once the food has been acted upon by the enzymes, it is then passed to the next section of the small intestine, the ileum. Digestion in the ileum. The ileum is the lower part of the small intestine and the longest section of the alimentary canal. It is largely coiled to fit into the limited abdominal space. The inner walls of the ileum contain secretory cells, some of which secrete mucus and others secrete an alkaline fluid known as intestinal juice or succus entericus. The arrival of chyme in the ileum stimulates the secretion of intestinal juice. The juice contains 4 enzymes that carry out digestive functions as follows. Maltase speeds up the breakdown of maltose to glucose. Sucrase speeds up the breakdown of sucrose into glucose and fructose. Maltase and sucrase complete the digestion of carbohydrates. Peptidase breaks down peptides into amino acids, and lipase, which breaks down lipids into fatty acids and glycerol. The process of digestion thus ends in the ileum, and the resulting watery emulsion of food is called chyle and contains soluble products of digestion ready for absorption. The entire canal is lined with an epithelial membrane that contains goblet cells, which secrete mucus. The mucus allows for smooth movement of food materials and also coats the wall of the alimentary canal, thus protecting it from being digested by enzymes. The next phase is absorption, and this is the process by which soluble products of digestion diffuse into the cellular lining of the villi. Diffusion of these products continues through the thin walls of the capillary network and into the bloodstream. Absorption of alcohol, some water-soluble vitamins B and C, and water-soluble salts take place in the stomach. Most absorption of final products of digestion occurs in the small intestine. The ileum is where absorption of digested food substances occur and is adapted to its functions in the following ways. It is long and therefore provides a large surface area for absorption. In addition, it is narrow so as to bring digested food into close contact with the walls of the ileum for easy absorption. The ileum is highly coiled in order to slow down movement of food and thus allowing more time for digestion and absorption to take place. It also increases the surface area for digestion and absorption. The inner lining of the ileum has large number of villi and microvilli, which increase the surface area for absorption of end products of digestion. The presence of a thin layer of cells through which digested food diffuses, the presence of a dense network of blood capillaries in the villi into which amino acids, sugars, vitamins, mineral salts, some fatty acids and glycerol are absorbed, and this helps to maintain a steep diffusion gradient. Finally, the presence of lacteoles in the villi for absorption of fatty acids and glycerol. Now, the molecules of amino acids and glucose pass through the epithelia of the villi and the capillary walls into the blood system by active transport. The capillaries drain into the hepatic portal vein where amino acids and glucose are carried to the liver before being allowed to join the general body circulation. Fatty acids and glycerol are absorbed into lacteoles of the villi, which drain into the lymphatic vessels. The lacteoles content join the general lymphatic system, which ultimately finds its way into the blood circulatory system, and the fatty acids and glycerol are then distributed to all parts of the body. Ingestion. The undigested and indigestible food substances reach the large intestine by the process of peristalsis. As these materials pass through the colon, water is extensively absorbed. The remaining semi-solid waste material is then passed to the rectum, where it is eventually eliminated from the body as fecal waste through the anus as the anal sphincter muscles relax. The cecum and the appendix have no function in humans. However, in herbivores, they contain a large number of bacteria which secrete enzyme cellulase that digest cellulose, breaking it down into glucose. The bacteria are in a symbiotic relationship with the herbivores. Some of the glucose is used by the bacteria, but most of it is assimilated by the herbivore. Assimilation. Now, the absorbed food products of digestion are carried into the bloodstream to the rest of the body. From the blood, the body cells are able to obtain these substances and use them in their metabolism. The incorporation of these products of digestion into the cell metabolism is known as assimilation. The different food types are assimilated and utilized as follows. Glucose is oxidized to release chemical energy during respiration, and excess of it is stored in the body as fats underneath the skin or in form of glycogen in liver or muscle cells. Fatty acids and glycerol are also oxidized to release energy, but most of the fatty acids and glycerol combine into neutral fats, which are stored underneath the skin to insulate the body against cold. Amino acids are used in the synthesis of proteins for general body growth and repair of worn-out tissues. In fats and glucose, they may be used to release energy through oxidation, which occurs in cases of extreme starvation. Vitamins. Vitamins are organic chemical compounds that are essential for a healthy body. They are obtained from fruits and vegetables. Some are synthesized in the body by the activity of microorganisms present in the gut. An example of this is vitamin K. Vitamins are destroyed when the food is excessively cooked. Although vitamins are required in small quantities, they play an important role in metabolic reactions. Some of them act as coenzymes in enzyme activities, while others influence intakes of certain substances into the body. For example, vitamin D influences the uptake of calcium ions in the gut. Such vitamins are not affected by the reactions they influence, and hence they can be used repeatedly. Lack of vitamins in the diet results in a breakdown of normal body activities and produce deficiency diseases. The breakdown can be corrected by including the necessary vitamins in the diet.
Without many of these discoveries and inventions, few of us would be alive today. Welcome to WatchMojo.com, and today we're counting down our picks for the top 10 greatest medical breakthroughs in history. Number 7. Painkillers. In the 1800s, two of the most important analgesics were introduced to the world, morphine and aspirin. Since then, the pain-killing properties of the naturally occurring chemical compound known as morphine have lessened the suffering of an immeasurable number of human beings and have led to the development of many other analgesics. Meanwhile, German chemist Felix Hoffmann's synthesis of acetylsalicylic acid into aspirin for medical purposes has turned the drug into one of the planet's most popular medications for pain relief.